development encompasses three stages from conception to birth. First, the germinal stage, the embryonic stage, and the fetal stage. First, germinal stage or zygote. Fertilization begins with the joining of a sperm and egg to form a zygote which travel to the uterus. Fertilization is a process in which a sperm cell from a male joins with an egg cell from a female to the form of zygote. This newly formed zygote then travels down the fallopian tube to the uterus, where it can implant and develop into an embryo. This marks the beginning of a new organism development. Cell division within 24 to 36 hours, the zygote undergoes rapid cell division. Cell division refers to the process by which the zygote rapidly divides into multiple cells within 24 to 36 hours after fertilization. This series of divisions, known as cleavage, transforms the single-celled zygote into a multicellular structure, paving the way for the further development into an embryo. Differentiation. Cells begin to differentiate into outer cells or placenta and inner cells or embryo. Differentiation is the process where cells develop into distinct types with specific functions. After several rounds of cell division, the cells of the developing embryo start to specialize, some from the outer layer, which will become the placenta that supports the growing embryo, while others become the inner cells that develop into the embryo itself. Blastocyst development. After about a week, the zygote matures into blastocyst with three layers: ectoderm, endoderm, mesoderm. After about a week following fertilization, the zygote develops into blastocyst, a structure that consists of three distinct layers: exoderm, endoderm, and mesoderm. Each layer will eventually give rise to different tissues and organs in the developing embryo. The ectoderm forms the skin nervous system. The endoderm becomes the linings of internal organs, and the mesoderm develops into muscles, bones, and the circulatory system. This stage is crucial for establishing the groundwork for embryonic development. The last one in the germinal stage, implantation. The blastocyst implants into the uterine wall, establishing a connection for nourishment. Implantation is the process by which the blastocyst, a fluid-filled structure formed from the developing embryo, attaches itself to the uterine wall. This connection is vital as it allows for the exchange for nutrients or oxygens from the mother to the embryo, establishing the foundation for further development and growth during pregnancy. Second, the embryonic stage or embryo. First, identification begins in the third week after conception when the cell mass is recognized as an embryo. Identification refers to the developmental stage that begins in the third week after conception when the rapidly dividing cell mass is recognized as an embryo. During this time, the cells begin to recognize and differentiate into specific structures and systems, marking the transition from a simple mass of cells to a more complex embryonic form. Second, nuchal tube formation, critical for brain and spinal cord development, occurring around 4 weeks. Neural tube formation is a crucial developmental process during embryogenesis, occurring around the 4th week of pregnancy. It involves the transformation of a flat sheet of ectodermal tube, which eventually gives rise to the brain and spinal cord. Third one, body structure development. By the 4th week, basic body structures emerge, including the heart and limbs by the 8th week. During the 4th week of embryo development, foundational body structures begin to form with notable developments, including the early formation of the heart and beginnings of lymph buds. The last one, neuronal development, neurons from around day 42 establishing early neural networks. Neuronal development begins around day 42 of embryonic growth, When neurons start to form and establish early neural networks, this process is critical for the formation of the nervous system, allowing for the communication between different parts of the body. The third stage, the fetal stage or fetus. Transition to fetus begins at 9 week marked by significant growth and brain development. The transition to the fetal stage begins at 9 week of pregnancy, characterized by significant growth and brain development. During this period, the fetus undergoes rapid physical change, with vital organs continuing to form and mature. The brain develops more complex structures, supporting essential functions and laying and groundwork for coordination and sensing the environment. Nervous system maturation. Continued development of the brain and spinal cord with reflexes appearing by the 12th week. During nervous system maturation, significant development of the brain and spinal cord occurs, leading to increased complexity and functionality. By the 12th week of embryonic development, basic reflexes begin to appear, indicating the time nervous system is becoming responsive and capable of coordinating simple movement. Sex organ differentiation. By the third month, reproductive organs develop distinct features. By the third month of pregnancy, or sex organ differentiation occurs, leading to the development of distinct reproductive features in the fetus. This process involves the activation of specific genes and the hormones that guide the formation for a male or female reproductive structures, such as the testes or ovaries, respectively. Growth and organ development. Significantly physical growth occurs in the second trimesters, with the fetus increasing in size and weight. During the second trimester of pregnancy, significant growth and organ development take place. The fetus experiences a marked increase in the size and weight, with body system becoming more functional. The last one, preparation for birth. From 7 months on, the fetus prepares for life outside the womb, with the lungs maturing for respiration. From 7 months or onward, the fetus begins preparing for life outside the womb, with crucial development occurring in various systems.